So with this video is showing in a very nice way the strategy that we as molecular biologists use to clone DNA. So in that case, if we want to study certain proteins or we want just to amplify the DNA, what we do is to use a vector, as been seen here. This vector have the characteristic that being able to reproduce, in this case in bacteria, but there are some vectors that are able to reproduce in insect cells or mammalian cells, so we can use different systems to do this expression. And what we can do is insert the DNA of our interest, because we know exactly how the nucleotides codify for proteins, so we can build up, depend on the amino acids of the proteins that we want to characterize, we can build up the DNA and try to clone it and to express it. So we insert that DNA into the vector, becoming a plasmid, and that plasmid can be expressed and amplified in bacteria and in other systems, and we can use it as well to explore the function of protein. So this is a very powerful, cloning DNA is a very powerful tool that we use here in molecular biology to identify the function of the proteins. There is another technique that allows us as well to not only to identify but to amplify the DNA, and that's called a polymerase chain reaction, or PCR for short. So the basic here with this reaction is to generate a cycles of reaction with enzymatic reactions that is going to amplify specific DNAs. So we have the DNA, as we have been talking all this time, double helix DNA, which is bound with different nucleotides, and they form pair base, and they form these alpha helices, the DNA helix. And what we need for the PCR reaction or the PCR components are the DNA, which is the template. We're going to use the primers. So these primers are small oligos, or small fragments of DNA. They are typically between 15 30 nucleotides that contain the specific sequence of the gene that we need to amplify. As you know, every gene has a specific sequence, but if we design a small fragment of DNA with a specific sequence that we have, we will be able to amplify just the DNA that that we require. For example, and I'm going to show you in the next slide, for COVID-19, this is what I have been using. So there is already the gene or the DNA sequence of COVID-19 has been identified. And to be able to see if a patient is positive for COVID-19, we take a sample. Well, I don't, but the nurses do. They take saliva, or they take a sample from the patient that contains the DNA, and they had already designed specific primers that are aiming to target the COVID-19 DNA, and they prepare this PCR reaction to be able to amplify the DNA. If the patient shows that he has COVID-19, that means that the patient is positive for COVID-19. But if there is no amplification of the DNA using these primers, it means that the patient doesn't have COVID-19. So this is what is being used actually with this pandemic around the globe. So these are the components, so the primers that are specific, the nucleotides that are the single nucleotides that the polymerase are going to use to build up a DNA chain, and the buffer that requires a specific cofactors for the enzyme to work and a specific pH. And of course, all these reactants are going to be placed in a tube, and the tubes is going to be placed in a thermocycle, that is the machine that is going to regenerate the program for PCR. The program basically is cycles, as I mentioned, with the temperature increases and decrease. So we have the double-strand DNA that we now know that is interacting by base pair. So this is the interaction of one strand and the other strand, and how we're gonna break this interaction and we're gonna separate this strand of the other is by increasing the temperature. When we increase the temperature, we are able to break the bonds that are of these interactions here from one strand to the other strand. And when we get single strands of RNA or DNA, then we lower the temperature and then we allow the primer, specific primers with the sequence that we want, to be interacting with the parent sequence of the DNA. So this is a primer that is in the reverse direction. This is the primer that goes in the forward direction. And then the enzyme, that is the polymerase, is going to bind to this site. I'm going to start copying the DNA, adding single nucleotides depending on the sequence of the DNA that you have there. So it's going to start adding nucleotides. The best temperature for this enzyme to work is 72 degrees. Depends on the enzyme that we are using. We can use different polymerases. In this case, it's Taq polymerase. And 72 degrees is the best for that. So the enzyme starts adding nucleotides and make a complete new strand of DNA based on this parent DNA or template. In this case, it's called template. And from one double strand DNA, we get 1, 2, 3, 4. And in the next cycle, this is going to open and it's going to generate 2 and 2. This is going to open and it's going to generate 2 and 2. And what we do this is the process in cycles. At least what I do my cloning, I usually do 30 cycles. So you can imagine the amount of DNA that I'm going to amplify in a very exponential way here to be able to obtain the desired DNA of interest, no? So this is, as I mentioned, something that has been lately in these days done but for the COVID-19. So when they increase the amount of DNA, they will be able to detect it with the system. So that's why they have to repeat these cycles many times to be able to see a signal, because sometimes the amount of DNA that is produced is not high, so the detection system is not going to be able to see it. So we repeat several cycles to be able to increase the amount of DNA, and the specificity of the system is given by the primers, because we're going to design the primers that are specific for the DNA that we want to amplify. So this is a very nice technique that we use in molecular biology for the identification and for the amplification of DNA in the systems. And this is the slide that I had prepared for you, because that's exactly what happened now with the COVID-19, as I mentioned, is a PCR reaction. So now, if someone asks you, you already know how is the process that is used for identification of COVID-19. And actually, the PCR diagnosis or test is very sensitive. And from a little bit of sample of DNA that you acquire because you amplified in cycles, you will be able to have a signal. If you don't have a signal, the probability that is negative is very high. Okay, so once we have all these techniques that we have been going through, and actually this knowledge we have been seeing so far, the DNA, was the composition of the DNA, the nucleotides, how the nucleotides interact to form a hybrid DNA or double-strand DNA, how this double-strand DNA can replicate or duplicate in the cells to have more and available for the function of the cells, as well how this DNA is transformed into RNA by a process that is called transcription, and how this specific RNA is transformed into a protein by a process that is called named translation. Additionally, we see how we are able to manipulate the DNA to be able to incorporate it in a vector or in a plasmid to amplify or to be able to study this DNA by the process that is called cloning DNA, to put it in a plasmid and express it in bacteria. And finally, we saw another technique that is for the amplification of DNA, to be able to increase the amount of DNA that we have, and to be able to identify that a specific DNA that we want to see is present in the sample.